Here, we have a team passionate to address mental health issue. What we want to do is to build a device that can allow quantitative diagnosis. So that's what we called the MANTATE, and also MANTATE team. Globally, 1 in 5 people is suffering from depression and anxiety. And currently, we're spending more than 2.5 trillion per year on mental disorder-related issues. But the field is still suffering from minimum understanding. The gold standard for diagnosis is using this Bible shown here, which summarizes all the symptoms related to any mental health disorder that can be found. But there's no single biological test in this book. There's no single test using information from blood, information from our brain, or even heart rate. And this is the state of art. And the gold standard, even in today, is based on self-report. Frequently, patients either don't have the motivation or it's just too complicated to describe their symptom and end up not reporting. So can you imagine if today, to treat diabetes without using glucose testing to know the blood sugar level? But this is exactly what mental health diagnosis is at today. So a year ago, we put together a team with PIs from 6 different departments and 2 different schools, with involvement of collaborators from business and the law school. And we have 5 different aims that we plan to use to address issues related to quantifying mental states to provide a quantitative diagnosis method. Of course, like any collaborative team putting together and working together for the first time, we encounter many challenges in trying to understand each other's language, try to understand each other's brain frames, thought frames. And through many conversations, meetings, and even repeating the same questions again and again, we start to learn from each other and start to really understand what the problems are and what the needs are. And to deepen our collaboration among the team members, we also have gone through a number of different exercises to build the team, to gel the team together. For example, presenting at the conferences and organizing a design workshop for mental health wearable design, and also working together to build prototypes. So in short, what we learned is that our initial goal was too ambitious. We tried to solve all problems, but it's not possible within the short time frame we have. And based on the feedback of the reviewers and the seed funding, we decided to greatly narrow the scope. And also, to the surprise of the engineers, we learned that we're not just diagnosing normal versus depression. We're actually trying to diagnose the exact type of depression, which Lee is going to elaborate next. Through talking to many people, from the patients, from people in industry, from venture capitalists, we understand that what we are building, there's a huge need for such kind of device. And if we're able to build it, this platform is going to be useful for addressing many problems related to mental health. But of course, we cannot solve all problems. So this Catalyst project is really a platform to allow us to better understand the problem, to define our goals, and also to build a community. And there's an urgent need to solve this issue. every 40 seconds, there's one person committing suicide because of mental health issue.
So we are extremely fortunate to be able to catalyze the multiple disciplines that Zhenan talked about in this project. And we can think of it as bringing together broadly two forms of circuits. If we're going to diagnose objectively conditions such as depression, we actually need to bring the biology to the table. And that would be similar to thinking of how do we currently diagnose cancer. We don't think of it as one broad category and think that that's going to be a sufficiently precise diagnosis necessary for interventions. We think of biologically based subtypes. Exactly the same in analogous form is how we're approaching mental disorder with a focus on depression, given the urgent public need. And here we are fortunate to have an advantage of an already established here at Stanford biologically based taxonomy, which takes a concept of how the human brain is functioning in terms of large-scale circuits necessary for human functions, so functions such as how do you reflect on your own thoughts, how do you regulate your emotions, how do you motivate yourself. And these dysfunctions in these different large-scale circuits give us types of depression that we observe in everyday behavior. So let me mention a couple of them. So the one on the left in blue, they're regions of the brain that connect together when you're freely associating or freely thinking, freely reflecting. And we do that when we're not focusing on a particular task. We do it when we're trying to relax. That part of the brain naturally forms that triangular shape, and we can observe it using an MRI scan. In depression, or one form of depression, we see that overly connected. The brain essentially gets stuck. And what that means is you start to brood or ruminate and worry. Another form of depression is the one in green, which is a part of the brain that is like your natural radar. It's picking up what's salient internally and externally. It creates a different form of depression that often creates avoidance of cues that may feel too salient and create anxiety. So with these types, we call them biotypes because they have a basis in brain circuits that we can assess with MRI. With these biotypes, we can then connect to another circuit, which is a circuit that's included in Mentate. So that gives us a way to measure mental states. So our first aim has been to look at the parameters that we establish with our biotypes and connect them to the measures with our Mentate. And what we have already in the first year is a set of coherent relationships that distinguish the biotypes in terms of the Mentate measures. So one example would be, if you just look at the blue type that I talked about, you can see it has very low levels of cortisol but high levels of change in heart rate, whereas the green one has high levels of cortisol along with high levels of heart rate. So we have 4 measures to do with skin conductance, heart rate, cortisol, and heart rate variability, and then the change in those under stimulus conditions that give us a way to differentiate these types. Let me hand back to Zhenan about how to do the other aspect of the circuit.
So now that we know there are several parameters that are directly correlated to the biotypes of depression, we went ahead to start building a Mentate that can measure these parameters. So through many iterations, now we have our first generation of working Mentate that incorporate a number of parameters shown on the bottom. They are all working ones. The one in the middle, heart rate variability and skin conductance, these, even though commercial devices are available, but we found that they either are very bulky or consume a lot of power, or the standalone medical version are very expensive. So we went ahead and built our own version, and now we found that still the motion artifact still needs to be addressed in these devices, in our own device. Also, our project has gathered interest from major companies. Apple now is sponsoring a number of Apple Watch to allow us to have the Landay to directly talk to Apple Watch so that we have the user interface can be built into the watch. DuPont is sponsoring to build a large number of the Landay and the first version is shown here, which is a flexible version of the Landay. The most challenging sensing that we found is to sense cortisol in the sweat. But we also made great progress in the past year. Now we have a platform, flexible transistor-based readout device that is highly durable under sweat condition. And we have also been able to find a competitive assay based on using the antibody attached to the surface, and then with the cortisol showing as red, when they are generated in sweat, they will kick off the antibody that's on the surface of the device and generate a large electrical signal. And this assay was found to have a detection limit in the nanogram per ml range, which is needed for sweat detection of cortisol. Also, it's insensitive to pH levels, which is important because our sweat pH changes when we exercise versus resting. Aim 3 is to preliminarily validate the Landay. We want to be able to, in the clinic setting, to show that we can differentiate two different types of depression. And also in the field, we want to show that our Landay can at least collect the 3 parameters that is needed to determine depression. So here we leverage the study that Lee's group already has undergo, where there are a large number of patients is being scanned to get their brain circuit connectivity to determine the exact biotype. So we're going to recruit those patients and test our Landay directly on these patients. And we have obtained the IRB approval, and the first clinical testing on patient has already been done. This is showing Jyoti, our clinic coordinator, is doing the first testing on the patient. In this task, we found, unlike the first 2 aims, where only a subset of the PIs were involved and working on the design of the devices or on the experiments, but in this aim, it requires all the PIs to participate, and there wasn't a clear design requirements or testing protocol existing for such devices. So the design team in the project really served as the glue to take the understanding from different team members and really align our project requirements for the final design to allow us to achieve the mandate. And through working together, we're very happy that our first version of mandate is being tested on patient. We also were able to perform our initial field testing just to get an idea of what are the current limitations of devices and what we need to improve. Of course, the first thing we learned is users may not want to even wear the device the way we designed them for, that is for the wrist. The first user put the device on the ankle. And we also learned the batteries didn't last long enough, and people exercise and have many activities, so the device need to be a lot more robust than what we currently have. In the next 2 years, our plan is to be able to have the mandate to show that we are able to differentiate 2 different types of subtypes of depression. And we want to show that the mandate can measure the parameters in the field. And also the Catalyst project is really a seed to allow us to build a community that can address mental health issues. So we want to continue the design workshops, hackathon activities, and also we want to use a mental health symposium to engage experts from law, from business, from ethics issues to discuss these important issues that we also have to be aware of. So in summary, thanks to the Catalyst project, it really brought together a group of us who have never worked together but passionate about mental health issue. And we learned that interdisciplinary work is challenging and exciting at the same time. But through alignment of goal, we're able to achieve a much more focused need to focus on the need to address critical issues. A year ago, we had the idea of building a mandate to address mental health issue. And now, a year later, we know much better what are needed, what can be done, and what still need to be done. We want to thank all the people who participated, the faculty members, undergrad students, graduate students, and postdocs. Many people volunteered their time, volunteered their ideas. We thank their participation.
We'd also like to highlight the scope and scale of impact of this project. It has been a challenge and a very exciting goal to bring together something that's tangible, concrete, quantifiable, but also can potentially have scale of impact for our nation and globally. And that is just to reinforce the scale of impact, which is the fact that currently every 40 seconds at least one person is dying from suicide. We have 405 million people with depression in our world. And currently in the US, it's the #1 cause of disability. Again, we'd like to acknowledge our collaborators and invite them to join us.
Thank you.
I have a question for Lee. So are the various subdivisions, biotypes of depression static, or do they vary over time? And how do you characterize that with MRI and with the mentate?
Right, that's a great question. They are what I think of as dual trait types, as in you could think of as similar to blood pressure, where it's conferring some natural variability, and that can fluctuate out over time, but it is trait-like in each person. So it has a conferring natural variability, but also acting as a risk factor at the same time that can shift into a zone of illness. What we didn't have time to mention is that we do have studies ongoing in the lab that actually serially scan people to look at how these biotypes change or don't change, and they are up to 80% stable over time in terms of the trait-like aspects, but are malleable in terms of intervention.
Okay, is there another question? Yeah.
So first of all, I admire that you got up and said that you needed to narrow your scope, because not everybody is willing to admit that. And so thank you for being honest about that. That's great. But at the same time, my eye was caught on one bullet that said a lot of what you're doing could translate fairly easily to other things, not just depression, but also basic stress and anxiety. Can you say a little more about that and what leap would be needed, or is it a leap or is it incremental to capture those other aspects?
I'll start and maybe you can add. So for a lot of other mental disorder or even just simple stress and anxiety, the three parameters that we're trying to measure here, heart rate variability, skin conductance, and cortisol, frequently have been used in the lab to perform analysis. But still, currently, a quantitative correlation has not been developed, especially a longitudinal correlation has not been developed.
To add on that, I'd say we can think of this like a prototype that would actually accelerate application to other areas. And from the brain side, that is because the circuits that we're looking at in the human brain are ones that are dysfunctional in other ways, in other conditions. So you could immediately think of anxiety, substance use disorders, which we had intended to look at. You can consider expanding to kids. You can consider expanding to stress disorders fairly readily.
And would that be just a matter of taking the data you're collecting anyway and doing more analysis on it, or is it more complex than that?
It's a little bit of both. So we can, when we acquire depression, we purposefully also look at anxiety. We do need to make a decision about ruling in or ruling out substance use, because it will impact the brain differently. But that can certainly be accelerated.
Okay, well, I think to keep on schedule, thank you very much for all your great work.